Good evening, everyone. I'm Isabel from the College of Medicine, Department of Molecular Genetics and Microbiology, and I'll be presenting my project MHV68 microRNA targeting the host AKT signaling pathway. Gamma herpes viruses are double-stranded DNA viruses that include human pathogens EBV and KSHV. And although it's difficult to study these human viruses, we can use a genetically related virus, MHV68, for our studies. Ultimately, we want to look at how gamma herpes viruses utilize their microRNAs to regulate B cell proliferation, specifically through the targeting of the AKT pathway. Generally, gamma herpes viruses will infect epithelial cells through lytic replication, and they then can go on to infect B cells, and this is where they establish latency. An important component of gamma herpes virus pathogenesis is their ability to encode microRNAs. These viral microRNAs can target host messenger RNAs and can either degrade them or repress translation of host proteins. So to identify these viral microRNAs, we did what is called Q-clash, or quick cross-linking ligation and sequencing of hybrids. And we were able to retrieve about 2,500 host messenger RNAs in MHV68 and compiled a list of targets of EBV and KSHV targets as well. Knowing we wanted to look at the AKT pathway, we looked at specific genes within this pathway and then looked at other genes that specifically affect AKT stability and activation. Here they're mostly kinases or phosphatases. When I analyzed all of our targets across MHV68, EBV, and KSHV, I found that over 1,500 of them are shared among all three viruses. To see where MHV68 targets appeared in several cellular pathways, I ran a pathway analysis, and Figure 5 shows you the top 15 targeted pathways of MHV68's viral microRNAs. Down here in Figure 6, you can see all three viruses' targets within this pathway, namely AKT, PP2A, PI3K, PDK1, and SHIP. So we know what host messenger RNAs are targeted by these viral microRNAs, but how can we assess the effects of these microRNAs in host cells? Well, to do that, I cloned in a region of MHV68's genome encoding its microRNAs, named the TEMA region, into a plasmid, and then transfected both fibroblasts and B cells with this plasmid. From there, I selected and expanded transfected cells into a polyclonal cell line. From here, I want to analyze host messenger RNA levels using qPCR in both cell lines of the transcripts listed in Figure 3b, and I also want to look at protein expression through Western blotting. I also would like to assess cell proliferation using a WST1 assay. And then in addition to the qPCR, I want to send off samples for RNA-seq to determine host messenger RNA levels. I would like to thank Dr. Tibbetts for his guidance and all other members of the Tibbetts Lab. Please, if you have any questions, feel free to reach out. Thanks for tuning in.